Hello students and welcome to the last session for this course officially Basics of Biology BT02 NOC 25. This is an extra class as demanded by the NPTEL authorities, and we will be solving questions from all over the questions from all over the lectures and assignments that we solved. One question from each as a summary of all the throughout the course that we have learned, and this will be a short session about 10 to 11 questions, and we will follow the same arrangement as we have followed throughout this course, rather happening on a Saturday. It is scheduled today on 15th April 3 p.m. to 5 p.m. So we will start this by thanking everyone who have joined this course, everyone who attended, gave their time for their live sessions, who watch these recorded lectures, who participate in the assignments, most importantly, and also the NPTEL authorities as well as Professor Vishal Trivedi for recording the lectures for this course. So once again, my name is Ashutosh Behra and I am a PhD scholar, PMRF scholar from IIT Kanpur. So along with me, there are other TAs for this course to help you assist in understanding this course better. Okay, so the format of the this class would be the same. I have prepared a presentation with 10 to 11 questions for all of you guys. These are multiple choice, single answer correct question, and we will be dealing with them one by one. I will post the questions on your screen. Read out the question loud along with the options. You have to tell me what do you think is the correct answer to this question, and then we will move ahead and look at what is the correct answer to this question, and then understand the concept behind uh, whatever the question is posing. Thus, clearing our doubts and having a practice of what we will be facing in our examinations and in quizzes. Okay, and I would request everyone who is attending the live session to please turn off your mic and video chat while communicating with me through the chat box only. I will be monitoring the chat box regularly for your answers, for your queries. If you have any questions, any doubts, so this is the moment you have to take courage and ask it. Otherwise, your doubt may remain unsolved. So please ask me questions. Please participate in this interactive TA session, and let's begin the presentation. Okay, so I will just share my screen shortly with you guys and ask for confirmation. Let me present my screen. This and open my presentation. Oops. Yeah, I hope you guys can see the screen properly and you can see the slides changing. If yes, then just let me know in the chat box whether you can see, and hear me. Okay, very cool. So once again, hi everyone, welcome to the extra class last session of this course, Basics of Biology NOC 25 BT02. This course is taught by Professor Vishal Trivedi at IIT Guwahati, and I am the teaching assistant for this course. My name is Ashutosh Behra and I'm a PhD student, PMRF scholar at IIT Kanpur. And this course is brought to you by NPTEL, and NPTEL authorities will be sharing this recorded video as well as the slides for your reference. And let's start this last week class with a word of note to everyone that all the best for your examinations, and I hope you do very well, and I hope you have taken home something fruitful of these out of these live TA sessions and the course that you are studying. Okay, with that, the first question of this session is on your screen. The frequently activated death protease that catalyzes specific cleavage of many key cellular proteins is dashed. It's a long question. The frequently activated death protease that catalyzes specific cleavage of many key cellular proteins is dash. Option A, thermolysin. Option B, ADAM 12. Option C, caspase 3. Option D, elastase. So you guys have to figure out which lecture module this question is and what is the question asking about. The frequently activated death proteases that catalyze specific cleavage of many key cellular proteins. So here is the hint. It is talking about death proteases. Does it ring a bell? Yeah. So that is the hint for you guys. And there are four options: thermolysin, ADAM 12, caspase 3, elastase. Let's take a minute and find out what is the correct answer. Tell me what you think is the correct answer, and I will reveal the correct answer in a minute or so. Okay, I think it's enough time for this question. The frequently activated death protease that catalyzes specific cleavage of many key cellular proteins is, and the correct answer to this question is option C, caspase 3. Caspase 3 is an important death protease associated with cell death pathway known as apoptosis. So does it ring a bell? Apoptosis and caspase 3. Caspase 3 is also known as the executioner caspase. Now let us look at apoptosis in detail. Apoptosis or programmed cell death (PCD) is a crucial biological process that eliminates damaged or unnecessary cells. So cell death is also essential as well as cell survival. It occurs through two main pathways, the intrinsic pathway and the extrinsic pathway. Both pathways ultimately trigger the activation of caspases, a family of proteases that degrade cellular components leading to death. And you know why caspases are known as caspases? Because they cleave, they are proteases, so they cleave proteins. Caspases cleave between aspartate acids, aspartic acid residues. Cysteine aspartic acid residue for C and ASP ases. So such cool naming convention you see. Caspases will cleave protein in between residues of cysteine and aspartic acid. leading to cell death. Now there are two important cell death pathway. One is the mitochondrial pathway, which is the intrinsic pathway, where the death signal comes from within the cell, and the extrinsic pathway, where the death signal is coming from outside of the cell. Right? Now what happens in intrinsic pathway? This pathway is activated by internal stress signals within the cell, such as DNA damage, oxidative stress, or lack of growth factors. So when a cell undergoes DNA damage or oxidative stress or mutations or chromosome instability, so all these internal events of the cell can trigger the intrinsic pathway of apoptosis. Cellular stress leads to release of cytochrome C from mitochondria into the cytosol. Cytochrome C binds to APAF1, forming the apoptosome, which then activates caspase 9 and initiator caspase. So you see, mitochondria sending in cytochrome C, which is then assembling APAF1 and the caspase 9 together to form what is known as the apoptosome, which then activates caspase 9. Activated caspase 9 triggers the activation of downstream executioner caspases such as caspase 3, caspase 6 and 7, which dismantle the cell by degrading proteins and DNA. So caspase 3 is important for intrinsic pathway of apoptosis. Now what is the extrinsic pathway or death receptor pathway? This pathway is initiated by signals from outside the cell, such as binding of ligands like TNF or FASL. FAS is the death ligand to death receptors on the cell surface. Like this, death receptors. If a death ligand binds to the death receptors, then what happens? Binding of the ligand to the death receptor triggers the formation of death inducing signaling complex or DISC. Just like we have RISC in siRNA, RNA induced silencing complex, here we have death inducing signaling complex DISC. The DISC activates caspase 8 and initiator caspase. Activated caspase 8 then activates downstream executioner caspases, similar to the intrinsic pathway leading to cell death. So here we have two types of caspases. One is the initiator caspase, which is activated in response to the external or internal signal, and then an executioner caspase, which is activated by the intrinsic initiator caspase and carried out the function. Now where does caspase 3 comes in? Caspase 3 is a crucial executioner protease that plays a central role in apoptosis or programmed cell death. So you see, initiator caspase is 289, executioner caspase is 367. And they are cut by initiator caspases and dimerize and then form the active enzyme. It is activated both during intrinsic and extrinsic apoptotic pathways and is responsible for cleaving wide array of cellular proteins, ultimately leading to characteristic morphological and biochemical changes of apoptosis. In essence, caspase 3 dismantles the cell, making it a hallmark of this cell death process. So that is why caspase 3 is an important executioner caspase in the apoptotic pathway. So now we have understood apoptosis in general, the two types of apoptotic pathway from intrinsic and extrinsic pathway that converge onto the executioner caspase 3. I hope it is clear to the students. If yes, then just let me know in the chat box with a yes or no. Very good. Okay, cool, good that now you have understood it. It will be easier for you guys to answer this question or any related question to apoptosis in the exam now. Okay, moving on to the second question of this session. The glycosidic bonds in starch is hydrolyzed by dashed. Another question for an enzyme. The glycosidic bonds in starch is hydrolyzed by dashed. Option A, trypsin. Option B, alpha amylase. Option C, mucin. Option D, pepsin. So there are 4 enzymes and the substrate is glycosidic bonds in the starch. Now, which of these enzyme can hydrolyze a glycosidic bond in the starch, or usually hydrolyzes the starch molecules at their glycosidic bond positions? Is it trypsin? Is it alpha amylase? Is it mucin or pepsin? Four options, and the question is clear to you. Let us take a minute from now and I'll reveal the correct answer. Meanwhile, keep posting your answers in the chat box. And on another notice, any new student who is trying to join this link will be allowed to this class. I will keep admitting them, people who are trying to request this. Okay, I think that's enough time for this question. The glycosidic bonds in starch is hydrolyzed by, and the correct answer to this question is option B, alpha amylase. Now, where is alpha amylase present? Alpha amylase is present in our saliva, which is also scientifically known as salivary amylase. Okay, major source of alpha amylase. Now, alpha amylase is an enzyme that catalyzes hydrolysis of the starch. This means it uses water to break down the long chains of glucose molecule that make up starch into smaller pieces. So it will break the starch into smaller pieces using water as one of the reactants. Random cleavage. Alpha amylase works by randomly cleaving the alpha 1,4 glycosidic bonds within the starch molecule. So now you know which molecule, which bond in the starch molecule is targeted by this enzyme. It's the alpha 1,4 glycosidic bond. So that is why this was the answer, that alpha amylase breaks the glycosidic bond present in the starch molecule. Now it is also an endohydrolase. Now what is an endohydrolase? Alpha amylase is an endohydrolase, meaning it can act on internal bonds within a large molecule rather than just on the ends. Just like we have another set of enzymes like restriction endonuclease. Restriction enzymes, they can cut from the end, some can cut from the middle, recognizing certain sequences like palindromic sequences. Here we have endohydrolase, which can act on internal bonds within a large molecule rather than just on the ends. It's not trimming from the ends, rather it can do its hydrolyzation in the internal bonds as well. Now what is the product of the action of alpha amylase? The breakdown of starch by alpha amylase results in formation of smaller oligosaccharides such as maltose and maltotriose and some dextrins. So the purpose of digestion is to break down complex organic molecules into simpler organic molecules, right? So that is what alpha amylase does. Alpha amylase will break the glucose into smaller oligosaccharides like maltose, maltotriose, and dextrin. These smaller sugars are then further broken down by other enzymes in the small intestine, ultimately releasing glucose for absorption. So, this is how glucose in the body is absorbed from starch. Now, what is the importance of this enzyme in digestion, in human digestion? Starch digestion is a crucial process as starch is a major source of carbohydrates in our diet. The action of alpha amylase is the first step in this process, and it is essential for the body to obtain the energy and nutrients it needs from starchy food. So, starch is the prime source of carbohydrates in our body. So, it is essential for the body to obtain glucose from starchy foods, and is begin this step of digestion is begun by none other than alpha amylase. On my right is a schematic showing the digestion of starch. The two important bonds, the alpha 1,4 linkage and the alpha 1,6 linkage. Amylase acting on the alpha 1,4 linkage, creating sucrose, maltose, isomaltose, dextrins as the oligosaccharides, which is then absorbed in the small intestine, transported, leading to further reduction in the glucose. Alpha amylase is an enzyme that plays a crucial role in breaking down starch into simpler sugars like maltose and glucose. It's found in both humans and animals and even in some microbes. So, not only humans and animals, it is also found in microbes. Where is alpha amylase found? So, the primary source is the saliva called salivary amylase or ptyalin. In the pancreas, which is called pancreatic alpha amylase, and in microbes, which is used for industrial purposes. Alpha amylase catalyzes the hydrolysis, breaking of alpha 1,4 glycosidic bonds in starch molecules, turning large polysaccharides into small absorbable sugars. So, this is what alpha amylase does, turning large polysaccharides into absorbable sugars. And on my right is a schematic showing the action of alpha amylase on the starch, breaking the 1 alpha 1,4 bond, creating maltose, maltriose, glucose oligomers, or dextrin. All this cleaves endo alpha 1,4 bond, and then there are other enzymes which leads to formation of the glucose. I hope it is clear to the students. We learnt a lot about alpha amylase, and I hope it helped students to understand. Very good. So with this we will move on to the next question. The next question is on your screen. The finger-like projections of small intestine that absorbs nutrients from the food is dashed. Finger-like projections of small intestine that absorbs nutrient from the food is dash. Option A, lacteal. Option B, arteriole. Option C, crypt of Libercoon. Option D, villi. Now this can be an easy question for you guys. It's just asking the name of microstructure, which is a finger-like projection of the small intestine that absorb nutrients from the food. This is a very famous structure, so you might recognize it right away after looking at the options. Let me take a minute and then come back to this question to reveal the correct answer of this question. Okay, I think this is enough time for this question. The finger like projections of the small intestine that absorb nutrient from the food is, and the correct answer to this question is option D, villus or villi. Villi are the finger like projections found in the walls of the small intestine, inner wall of the small intestine, that helps in absorbing nutrient from the food, isn't it? Now let us understand what is a villi and how does absorption happens through the villi and what are its adaptations for good adaptation, sorry, absorption. Villi are tiny finger-like projections that line the small intestine, significantly increasing its surface area. So this is the first line about villus. Villi is a structure which is a finger-like projection of the intestine, significantly increasing its surface area. This increased surface area aids in the absorption of nutrients from digested food into the bloodstream. So when there is more surface area, there is more absorption and releasing of nutrients into the bloodstream. They are crucial for efficient digestion and nutrient absorption. Who they? The 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 villi. So this is a diagram of the small intestine starting with the duodenum, the jejunum and the ileum. Plicae circularis, valves of the Crecking, which is there in the small intestine. Serosa, longitudinal muscle layer, circular muscle layer. And if you cut open through a single cross section like this, enlargement of the plicae circularis, there are these finger-like projections brushing the inner side, and these are known as villi or villus. And here is a detailed structure of a villus. There is a there is the intestinal epithelium, absorptive cells or enterocytes are present. There are some goblet cells which secrete mucus, endocrine cells, we will study about it, intestinal gland, Leverkusen gland, and the Paneth cells. Then there are blood capillaries. The lymph node also has a lacteal inside the villus. There are crypts of Leverkusen here and brush border cells with microvilli. Now, what is the comparison between a villus, crypts of Leverkusen, and lacteal? Location: villus lines the small intestine, while crypts of Leverkusen between the bases of the villi. So you can see it in the diagram. If this is villus, or if this white area is villus, then the underlying tissue, the 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 underlying crypts are known as crypts of Leverkusen between the bases of the villi. And lacteal, inside the core of each villus is a lacteal, is a lymphatic node. What are the functions of villus, crypts of Leverkusen and lacteal? Villus increases the surface area for nutrient absorption. Crypts of Leverkusen secretes intestinal juices, renews cells, protects, and lacteal absorbs dietary fats into the lymphatic system. Now, what are the key features of the villi? Villi have finger-like projections covered in covered in microvilli or brush border cells, gland-like pits with stem cells, goblet cells, Paneth cells are all present in the crypts of Leverkusen. And a lacteal is a lymphatic capillary transporting fat via the lymph. And here is a schematic showing all the cell types present in an intestinal lining. We have the villus, we have crypts of Leverkusen, we have Paneth cell, goblet cell, intestinal stem cells, transit amplifying cells, and enteroendocrine cells. All kinds of cells in the intestinal lining which help in various functions. I hope it is clear to the students that The finger-like projections in the small intestine which help in absorption of food and nutrients is known as villas or villi. Okay, cool. Coming to the fourth question, the casein protein in dairy products are digested in stomach by dashed. The casein protein in dairy products are digested in the stomach by dashed. Option A, chymotrypsin. Option B, trypsin. Option C, elastase. Option D, carboxypeptidase. Which of these are involved in digestion of dairy products inside our body? And the hint is already given in the question. The dairy product is rich in casein, which is the milk protein. Now, how does the body digest milk protein? Is it through chymotrypsin? Is it through trypsin or elastase or carboxypeptidase? Take a look at all the options. Take your time and let me know what is the correct answer to this question. I will reveal the correct answer in the end. Okay, I think that's enough time for this question. The casein protein in dairy products are digested in stomach by, and the correct answer to this question is option A, chymotrypsin. Option A, chymotrypsin is the correct answer. Chymotrypsin is the enzyme which supports in milk digestion or casein digestion in the stomach. Let's have a look at digestion of milk in the stomach and what is chymotrypsin. Chymotrypsin is not the main enzyme responsible for digesting milk. The main enzyme is renin. But it does play a supporting role in breaking down milk proteins. How? And particularly casein is acted upon by chymotrypsin. What is chymotrypsin? It is a protease enzyme, specifically a serine protease. Now, what are serine proteases? Serine protease can target proteasomal degradation, can target breakage of protein at serine residues. They make these serine, they use their serine residue as nucleophilic attack center for catalyzing the reaction. Now, what is the source of chymotrypsin? Chymotrypsin is secreted by the pancreas as inactive chymotrypsinogen and activated in the small intestine. So, at first, it is secreted as an inactive enzyme and then acted upon and activated in the small intestine, but it is released from the pancreas. Now, what is the function of chymotrypsin? It breaks down protein by cleaving peptide bonds, especially at aromatic amino acids like the phenylalanine, tryptophan, and tyrosine. So, this is an enzyme which will cleave most likely after these positions: phenylalanine, tryptophan, and tyrosine. Chymotrypsin and milk digestion. Milk composition. Major protein is casein. Major protein and other proteins are whey proteins. like lactalbumin and lactoglobulin. Casein is about 80% of the milk, and then the other proteins are lactalbumin and lactglobulin. These are whey proteins. Now, what is the role of chymotrypsin? After milk reaches the small intestine, chymotrypsin helps further break down casein, which was partially coagulated or digested in the stomach by renin or pepsin. This was the enzyme that I was talking about, the primary enzyme responsible for digesting milk. It cleaves specific peptide bonds in the casein structure, turning them into smaller peptides. These peptides are then further broken down by other enzymes like carboxypeptidases and aminopeptidases into amino acids which are absorbed. So if you see the mode of action of chymotrypsin, catalysis, this is the uncomplexed enzyme, this is the catalytic triad forming the histidine 57, D10 aspartic acid 102, and the serine 195 OH, which is the serine protease, which is also known as the area in which this OH, serine 195 OH, is situated is also known as oxyanion hole. So once the uncomplex enzyme forms a catalytic triad, it is activated by serine OH attack on the C double bond O of the amino acid peptide bond, and in the oxyanion hole it forms a tetrahedral intermediate, and then there is rearrangement inside the intermediate leading to formation of first product compound, first product. After the first product departs, an acyl enzyme is returned. Water binds and attacks the C double bond O, and then intermediate rearranges. After the rearrangement of intermediate, another second product disappears. I hope it is clear to the students. We have spent ample time about chymotrypsin and reading about how chymotrypsin helps in digestion of the milk. If yes, then let's move ahead with the fifth question. The deficiency of factor 9 during blood clotting results in dashed. I'll read out the question again. The deficiency of factor 9 during blood clotting results in dashed. Option A, von Willebrand disease. Option B, hemophilia C. Option C, hemophilia B. Option D, hereditary angioedema type 3.
Okay, I think that's enough time for this question. The deficiency of factor 9 during blood clotting results in, and the correct answer to this question is option C, hemophilia B. Deficiency of factor 9 leads to hemophilia B as a disease. Now, what is blood clotting? Blood clotting, also known as coagulation, is a vital process that stops bleeding when blood vessels are injured. It's a normal part of the healing, but can also be dangerous if it occurs in the wrong place or doesn't dissolve properly. Right? You almost have learned about the heart attacks. Heart attacks happen when the coronary artery is blocked or there is blood clot. Internal injury causes blood clot. How does blood clotting work? Damaged blood vessel cells release chemical signals. The signal causes the blood vessel to narrow and platelets to stick to the blood vessel. Blood clotting, also known as coagulation, is a vital process that stops bleeding when blood vessels are injured. It's a normal part of healing but can occur in the wrong place or can be dangerous if it occurs doesn't dissolve properly. Damaged blood vessels release chemical signals. The signals cause blood vessels to narrow and platelets to stick to the vessel. Platelets change shape and clump together to form a plug. Clotting factors include fibrin, form a mesh that traps platelets and blood vessels, forming a clot. Hemophilia B, also known as factor 9 deficiency or Christmas disease, is a hereditary bleeding disorder caused by deficiency or defect in clotting factor 9. This means the blood struggles to clot properly, leading to prolonged bleeding episodes. Hemophilia B is a genetic condition where the body does not produce enough or has a faulty version of factor 9, a protein vital for clotting. A mutation in the F9 gene, which provides instructions for making factor 9, is responsible for this disease. And people with hemophilia B may experience prolonged bleeding for injuries, excessive bruising, and in severe cases, bleeding into joints or muscles. I hope it is clear to the students. Very good. Moving on to the next question. The waves generated in a brain when a person is attentive and thinking actively is dash. The waves generated in the brain when a person is attentive and thinking is dash. Option A, beta waves. Option B, delta waves. Option C, theta waves. Option C, alpha waves. Which of these is released when a person is attentive and thinking actively? Okay, I think that's enough time for this question. The waves generated in a brain when a person is attentive and thinking actively is option A, beta waves. Beta waves are one of the many waves that are produced in the brain. There are gamma waves from frequency 30 to 100 hertz. And what are the function of these gamma waves? They help in insight, learning, and problem solving. Beta waves Beta waves frequency from 12 to 30 hertz, involved in active thinking, alertness, scrolling. Alpha waves from 8 to 12 hertz, which is released when there is relaxation, meditation, and lucid awareness. Theta waves 4 to 8 hertz, released while daydreaming, intuition, and creative thinking. And delta waves is released during deep sleep, recovery, and unconsciousness. Frequency 0.5 to 4 hertz. Beta waves are high frequency, low amplitude brain waves associated with state of wakefulness and active focused thinking. They are typically observed when individuals are alert, concentrating, and engaged in mental activities like problem solving or decision making. These are the key characteristics of beta waves. Beta waves range from 12 to 38 Hz cycles per second. Dominant state, they are the dominant brainwave pattern during normal waking consciousness and when actively engaged in mental tasks. Beta waves are associated with alertness, focus, concentration, and conscious thought. And beta wave can enhance focus on problem solving in the frontal lobe, supports active learning and alertness, facilitates critical thinking and focus in the parietal lobe, drives logical thinking and analysis. In the occipital lobe, associated with alertness and cognition, boosts cognitive processes and learning, and in the temporal lobe, supports critical thinking and focus, and supports analytical and decision making. I hope this is clear to the students. Moving on to the next question. The resting potential in neuron is attained by taking in dash. This will be an easy question. The resting potential in neuron is attained by taking in dash. Option A, chloride, Cl1 minus, Cl minus chloride ions. Option B, K plus, potassium ions. Option C, Ca2 plus, calcium ion. Or option D, Na plus, sodium ion. Which of these ion is taken in and the resting potential is achieved? Okay, I think it's time for me to reveal the correct answer to this question. The resting potential in neuron is attained by taking in potassium plus ions. K plus ions are the answer. K plus ions travel inside to make the resting potential of the neuron negative. Now, what is resting membrane potential? The membrane potential of an inactive or a resting neuron is the resting membrane potential. So, if you measure it, it will be minus 70 millivolts. And this is a neuronal membrane showing all different kinds of channels present on the membrane. There is a potassium channel, there is a Na plus K plus transporter or pump, and a sodium channel. And at the resting state, there is more amount of sodium present in the outside of the cell and more amount of potassium that is present inside of the cell. The resting potential of a neuron is the state negative electrical potential difference across the neuron's membrane when it is not actively transmitting a signal. It is typically around minus 70 millivolts, with the inside of the cell being more negative than the outside. This potential is maintained by a variety of mechanisms, including the sodium potassium pump and leak channels. And key points about neuron resting potential: the electrical difference between the inside and outside of a neuron when it is not firing an action potential. It is around minus 70 millivolts, that though it can vary slightly. And the sodium potassium pump actively transports sodium out and potassium in, maintaining the concentration gradient and thus the resting potential. Leak channels allow the passive movement of potassium out of the cell, further contributing to the negative charge. So this is how the resting membrane potential is maintained in a neuron which is not actively firing an action potential. Here on my right is an image for the sodium channel, sodium potassium pump, 3 sodium out and 2 potassium in from the extracellular milieu to the cytoplasm. Okay, moving on to the next question. The nerves of taste and saliva secretion originate from dashed. The nerves of taste and saliva secretion originate from dashed. Option A, midbrain. Option B, olfactory epithelium. Option C, pons varoli. Option D, medulla oblongata. The nerves which are responsible for taste and saliva secretion originate from midbrain, olfactory epithelium, pons varoli, or medulla oblongata. Let us take a minute and reach the correct answer to this question. Okay, time to know the correct answer to this question. The nerves of taste and saliva secretion originate from. Correct answer is medulla oblongata. Medulla oblongata is the point for involuntary control in our body, and the nerves of taste and saliva secretion originate from the medulla oblongata. So here is a diagram showing the important nerves for gustation, which is taste, olfaction, and taste and smell, are the cranial nerves 7, 9, and 10, the trigeminal maxillary branch, trigeminal ophthalmic branch, and trigeminal mandibular branch. The facial nerve, cranial nerve 7, primarily provides taste sensation to the anterior two-thirds of the tongue, while the glossopharyngeal nerve, cranial nerve 9, innervates the posterior third. The vagus nerve, cranial nerve 10, also contributes to taste perception in the root of the tongue and epiglottis. These nerves originate from the brainstem. So 7, 9 and 10, all of these nerves originate in the medulla oblongata, responsible for taste perception. For taste, there is facial nerve CN, cranial nerve 7, innervates the anterior two-thirds of the tongue, specifically through the chorda tympani branch, shown in the diagram here. Glossopharyngeal nerve C, cranial nerve 9, innervates the posterior one-third of the tongue, and vagus nerve, cranial nerve 10, contributes to taste sensation in the root of the tongue and epiglottis. And saliva, facial nerve 7, cranial nerve 7, provides parasympathetic innervation to submandibular and sublingual glands, and glossopharyngeal nerve 9 innervates the parotid and salivary glands. So all these nerves are responsible for saliva secretion as well as the taste receptors in the tongue are connected to the medulla oblongata by the cranial nerves 7, 9 and 10. I hope it is clear to the students. Moving on to the next question. Mineralocorticoids increases blood volume by increasing sodium water reabsorption and K plus excretion from dashed. Come on guys, this can be an easy question. Mineralocorticoids increase blood volume by increasing sodium water reabsorption and potassium excretion from dash. Option A, liver. Option B, pancreas. Option C, kidneys. Option D, large intestines. Which of these is acted upon by mineralocorticoids for increasing sodium water reabsorption and potassium excretion? Water reabsorption and excretion. These two keywords are enough hint for you guys to reach the correct answer to this question. Let us wait for a minute and then come back to this question for the correct answer. Okay, I think it's time to answer this question. Mineralocorticoids increase blood volume by increasing sodium water reabsorption at K plus excretion from the kidneys. Option C, kidneys, is the correct answer. What are the functions of the mineralocorticoids? And one of the prime example is being aldosterone. It increases the reabsorption of sodium from distal renal tubules and increases excretion of potassium from the distal renal tubules, excretion of H plus ions from the distal renal tubules, water reabsorption along with sodium ions. And sodium ion content of sweat, saliva, and other secretions is decreased when there is aldosterone secretion. Acts as a long term regulator of blood pressure. So, this is the function of the mineralocorticoid aldosterone. So, aldosterone primarily acts on the kidney, causing potassium reabsorption, H plus reabsorption, sodium reabsorption, which is thereby increasing plasma volume expansion, renal blood flow, peritubular capillary pressure, backflow of Na plus and H2O into the renal tubule, causing natriuresis or natural urine production. Plasma volume expansion also leads to stretching of cardiomyocytes and secretion of atrial natriuretic factor leading to natriuresis. So all these are the effects of the hormone aldosterone, which is a mineralocorticoid leading to retention of water in the kidneys along with sodium reabsorption and potassium ion excretion. Clear to the students? Yes, and moving on to the next question. The appearance of different species of Darwin's finches is due to dashed. Now changing gears, the appearance of different species of Darwin's finches is due to dashed. Option A, migration and adaptation to different location. Option B, artificial selection by chemical. Option C, gene cloning followed by selection. Option D, insertion of foreign gene. Which of these phenomena best explains different species of Darwin's finches? Let us take a minute and try to find out the correct answer to this question.
Okay, time for me to reveal the answer to this question. The appearance of different species of Darwin's finches is due to option A, migration and adaptation to different location. This is a this is a pie chart showing all the different variety of Darwin finches, starting from the vegetarian tree finch to the large ground finch. Depending on the food that they eat, they have characteristic physical adaptations, like parrot-like bill for fruit eaters, grasping bills for insect eaters, probing bills, warbler finch, cactus eaters, seed eaters have crushing bills, and there are variety of ground finches, tree finches, and so on. Darwin's finches exemplify adaptive radiation, a process where a single ancestral species diversifies into multiple species that occupy different ecological niches. This diversification occurs in response to varying environmental pressures, primarily different food sources, leading to beak shape and size variations that allow finches to exploit diverse resources. Darwin's finches and the Galapagos. Darwin observed that the finches on the Galapagos Islands exhibited remarkable beak diversity, such as each species adapted to a specific food source. From seed eating ancestor, the original finches were seed eaters, but upon colonizing the islands, they encountered new food sources and environmental conditions. And beak adaptation, the natural selection favored finches with beak shapes and sizes that were best suited to cracking hard seeds, probing insects, or consuming other available food. And this led to the evolution of multiple finch species, each with specialized beaks and feeding habits, effectively filling different ecological niches on the islands. I hope it is clear to the students. Yes. And the next question is on the screen. The end product obtained in a Stanley Miller's experiment is dashed. The end product obtained in Stanley Miller's experiment is option A, mixture of different proteins, option B, mixture of amino acids, option C, mixture of different types of DNA, option D, different types of RNA species. Which of the following was first obtained as an end product in Stanley Miller's experiment? Let us take some time and try to reach to the correct answer in a minute or so. Okay, time to answer this question. The end product obtained in Stanley Miller's experiment is mixture of amino acids, option B. It's a mixture of amino acid that was obtained in Stanley Miller's experiment. Now, what was Stanley Miller's experiment? Stanley Miller's experiment is one of the most famous studies in the field of origin of life's research. It provided the first evidence that organic molecules necessary for life could be formed from inorganic components under conditions thought to resemble those of early Earth. So, how did life originate in early Earth? For finding out that, They mixed water from the ocean, heating the water and the organic mixture. There are traps and cooled water containing organic compounds. There is a condenser and electric spark simulating lightning with all these gases: water vapor, methane, ammonia, and hydrogen. And what was each component representing? Gases: ammonia, methane, hydrogen, water vapor simulated Earth's primitive atmosphere. Boiling flask simulated the ocean. Water was heated to produce steam. Electrodes simulated lightning, sparked energy into the gas mixture, and there was a condenser to cool the gases back into liquid, which then dripped into a collection trap. And in the collection trap, collected the condensed water and new molecules formed. What was the purpose of the experiment? To test whether simple organic molecules like amino acids could form spontaneously from the gases believed to be present on primitive Earth, using energy sources like lightning. And these were the results. After a week, the experiment produced a variety of organic compounds, including amino acids. What is the significance of this experiment? The results provided strong support for the primordial soup theory, which suggests that life could have originated from simple chemical reactions in a primordial ocean. And the key findings of this study: the experiment successfully demonstrated that the abiotic synthesis of amino acids, which are essential for formation of proteins, is a crucial step in the development of life. And on my right is the famous Stanley Miller with his apparatus for the Stanley Miller's origin of life experiment. I hope it is clear to the students. If so, with this we have come to the end of our flurry of questions in for the extra class as well. I'll stop presenting here. Yeah, so with this we have come to the end of all the questions in the extra class. We have looked at basics of biology in a completely different perspective now. We have solved many questions, understood many concepts about the physiology of the animal, of the human body, the animal kingdom, cellular processes, and I hope these TA sessions had helped you in your study and answering your questions in the exam. Well, I have to end this call, and thank you for participating. Thank you for being with me in this journey. Hoping to see you, some of you, in another courses, because I'll be taking TA shift for the next semester as well. See you soon, and bye bye, and all the best for your examinations. I will end this call now. Thank you.